I'd like to thank the organizing committee for the opportunity and privilege to speak here today. Excuse me, I'm not sure which is more intimidating, this pointer that I've seen everyone had trouble with, or standing here and talking about 50-year-old drugs. The lower button. So basically, one of the research efforts in my laboratory and really are focused on the examination of the epigenetic effects of cigarette smoke and other carcinogens on the epigenome and how these mediate pluripotency and malignant transformation. We've done extended these to a number of translational clinical trials using chromatin remodeling agents to exploit the DNA methylation paradox by upregulating tumor antigens, particularly the cancer testis antigens, and to simultaneously restore tumor suppressor gene activity. We have novel cell-based vaccines that are in clinical trial, and we've most recently proposed and have CCR approval to use an oral DNA demethylating agent with a tumor lysate vaccine and nivolumab, which I'm not going to discuss today. I'm really going to focus on our efforts to resurrect mithromycin as a means to target stem cell signaling in thoracic malignancies. And the strategies that we've used to characterize epigenomic alterations in these malignancies include reprogramming of normal small airway epithelial cells to pluripotency. We have a number of in vitro tumor initiation and progression models where we're using cigarette smoke condensates and more recently water pipe condensates generated in our laboratory, and we have some in vivo progression models. And then all of our findings from these models are validated against surgical specimens from our lab. What we have seen over the years, and this is published data, so I'll move through it fairly quickly, is that there are very dynamic responses to chromatin in responses to cigarette smoke carcinogens. Within hours to sometimes days or weeks, we see cancer-associated histone alterations that are believed to be hallmarks of cancer, suggesting once again a very dynamic chromatin response. Over more extended exposures through this DNA methylation heat array, we see a progressive change in DNA methylation across normal bronchial epithelial cells, and this coincides with an upregulation or induction of an enhanced soft agar clonogenicity. When we evaluate these clones from cigarette smoke exposed cells relative to the few cells that grow out in soft agar from the control cells, we see a remarkable clonal appearance from cigarette smoke, which suggests a very strong selective pressure to establish an epigenetic signature in response to these carcinogens. What's basically emerged from all of this preclinical work are several central themes that basically demonstrate that cigarette smoke, either through direct engagement of polycomb machinery or through upregulation of oncogenic or oncomeres or inhibition of tumor suppressive microRNAs, are leading to upregulation of MYC and KRAS, activation of Wnt5a and non-canonical signaling through the antagonism of Wnt antagonists or repression of these. And what's interesting is that these changes all have been described as mediating a stem cell phenotype in pulmonary carcinomas. We extended these studies to try to evaluate more extensively the effect of cigarette smoke on pluripotency. This is just a flow cytometry study looking at the side population. And what we saw was that cigarette smoke was mediating a significant upregulation or expansion of the side population in lung cancer cells. We saw this phenomenon as well as in esophageal cancer cells, but this was not seen in normal small airway epithelial cells. This phenomenon actually coincided with upregulation of ABCG2, which is a xenobiotic pump protein and putative stem cell marker. What you see in the top panel are quantitative RT-PCR showing a dose-dependent increase in ABCG2 across several different cell lines. This phenomenon is really not seen in small airway epithelial cells. It's seen to some extent, but the magnitude is certainly not as great. These are the confirmatory immunoblots in the flow cytometry data. We further characterize the effects of cigarette smoke on ABCG2. This is a deletion mutant experiment in which we serially diluted several SP1 sites. And what we saw was an abrogation of the induction or basal expression of ABCG2 in this in vitro system. We tried to then recapitulate this using a drug that we might be able to take to clinical trials, and mithramycin was the one that came to mind. Mithramycin had been a drug that had been used in the 60s and early 70s, had mediated cancer complete responses actually in about 10% of patients with sarcomas and germ cell tumors, but had been disregarded or discarded because of excessive toxicities, which retrospectively have not been really well characterized. But what we saw was that mithramycin could not only inhibit basal expression of ABCG2, but completely abolish the induction of ABCG2 mediated by cigarette smoke. We then did some additional flow cytometry experiments which showed that mithramycin could reverse the or decrease the pluripotent side population, and a single 24-hour exposure of mithramycin in tissue culture dramatically mediated growth inhibition in lung and esophageal cancer cells. And furthermore, it mediated a dose-dependent repression or inhibition of growth of subcutaneous tumor xenografts in athymic nude mice without obvious systemic toxicity. And this was using clinical grade mithramycin. So on the basis of this, we then performed some additional gene array analysis, both in vitro and in vivo. We saw dramatic changes in response to mithramycin in a dose-dependent manner, and further higher order array analysis revealed that 8 of the top canonical pathways mediated or inhibited by mithramycin mediated stem cell signaling and pluripotency. And on the basis of this data, we then performed some additional confirmatory quantitative RT-PCR experiments, which showed that mithramycin was inhibiting a variety of epigenetic mediators, including DNA methyltransferases and a variety of core components of polycomb repressive complex, which many of you know is a critical mediator of stem cell signaling and aberrant tumor suppressor gene repression in a variety of cancers. On the basis of this, actually, the NCI contracted with a company in Israel to produce mithramycin. It was brought into DTP, quality controlled, and then formulated at the NCI for clinical trials. We conducted a phase 2 trial of patients in thoracic malignancies. There were 12 patients that were accrued to this study, including 6 patients with mesothelioma, 2 esophageal cancer patients, and 2 synovial cancer patients. We used the previous schedule of 7 daily infusions every 28 days at the previously approved dose. There were 20 cycles that were administered in 12 patients. All were evaluable for toxicities, and there were no objective responses that were observed in the 7 evaluable patients. Interestingly, despite all of the concerns about toxicity, we really saw none. We saw no neurotoxicities, no cardiac toxicities, no myelosuppression, but the 8 patients experienced transient and asymptomatic dose limiting transaminitis. It became fairly clear when in the patients that would develop this, we'd start to see an uptick around day 3 in the AST and the ALT, and by day 4, they clearly were in grade 3 or grade 4. And so we managed actually to get some biopsies on patients that appeared to be starting to have this. The biopsies were obtained on day 4, and the liver biopsies showed essentially apoptotic cell death in the liver. The question then became, well, is this related to drug levels in these patients that do and do not have the liver toxicities? And there were no PK data from any of the trials of mithramycin in the 60s and 70s. So we developed an assay at the NCI. And basically what we observed is that the peak and steady state concentrations were much lower than what we had predicted based on what limited data were available from animal studies. And there were no significant differences in the PK values in patients that did and did not have the hepatotoxicity. We then, because the differences were so striking, we used a DMET microarray analysis, which is a CLIA-certified array-based technique. There are about 2,000 SNPs involving about 230 enzymes involved in drug metabolism and transport. And what we observed was that the grade of hepatotoxicity actually correlated highly significantly with ABCG4 and ABCG11 polymorphisms. And both of these are pump proteins that are involved in bile flow. And this was then confirmed using hepatocytes from warm autopsies. And mithramycin appears to be a member of a rare class of chemicals that impedes sinusoidal blood flow and therefore can cause central vein toxicity. And it therefore becomes possible that what we're seeing the hepatotoxicity in are the patients that have the polymorphisms with lower levels of these pump proteins before treatment. Because we then used the modeling from our PK data, and we then could actually try to recapitulate the drug exposure conditions that we had seen in the preclinical animal models. The levels of mithramycin that we had seen that were mediating very dramatic growth inhibition were in the 50 to 100 nanomolar range, and we now actually can estimate the drug concentrations and the exposure conditions over a 24-hour period that will recapitulate this. And so this has led to a revision of our phase 2 trial, where there'll be a phase 1 component, which will basically establish the pharmacokinetics and the maximum tolerated dose of mithramycin administered over a 24-hour infusion every 2 weeks instead of every 28 days. And then the phase 2 component will be to evaluate the objective response rates. And in this trial, all patients are going to be selected based on pharmacogenomic profiling for a favorable ABCB4 and ABCB11 genotype. Now, if I can just spend the next couple of minutes talking specifically about this compound in mesothelioma. These are the growth inhibition curves following treatment of established pleural mesothelioma xenografts in athymic nude mice. These are two different cell lines that we've established, and you can clearly see very dramatic growth inhibition in subcutaneous tumor xenografts following intraperitoneal mithramycin administration. We then developed a carcinomatosis model in which mesothelioma cells were injected, and we established this model, following which we then began mithramycin injections intraperitoneally, and after about 3 weeks, 98% of the control mice died of fatal carcinomatosis, and we had actually cured about 70% of the mithramycin-treated mice. The remaining mice basically had minimal residual disease, and from a surgical perspective, if we had seen this in clinical settings, we would clearly go after this residual disease. So this was fairly encouraging. We've gone on to actually further characterize the mechanisms of growth inhibition in mesothelioma cells, and what we see is that mithramycin is mediating DNA damage, as shown here by the gamma H2AX increase in a dose-dependent manner. And by immunofluorescence and beta-gal assays and flow cytometry, we saw basically a senescent phenotype with a G0, G1 cell cycle arrest and the induction of some autophagy. What was interesting when we then performed some of the microarray analysis is that we saw that p53 signaling was emerging both in vitro as well as in vivo as a constant pathway. And we then went on to demonstrate that the mechanism of growth inhibition is attributable to a large extent to repression or depletion of SP1 and activation and increase of p53. And this becomes particularly relevant in the mesothelioma patients in which p53, for the most part, is wild type. And it actually could explain some of the rationale for trying to target p53 wild type tumors. We saw this both in vitro as well as in vivo. Interestingly, with the concomitant upregulation of p53, we saw upregulation of MDM2, which obviously can inhibit p53 function, and so this led us to see whether or not it would be reasonable to target p53 MDM2 interaction. Parenthetically, it may be that the sarcoma patients in which we saw almost very rapid increase in tumor growth following mithramycin may have been due to this upregulation of MDM2. And in the last couple of slides, I'll just demonstrate that we've looked at combinations of mithramycin with Rita, as well as a Roche compound that either activate wild type and mutant p53 or inhibit binding of p53 to MDM2. These are ApoBRDU assays demonstrating a fairly nice synergistic interaction of very low dose mithramycin with Rita. And these are growth assays in vivo using 1/3 of the intensity of mithramycin dosing with Rita here showing very nice additive effects here. And then when we do a similar in vivo experiment using the Roche compound, we have very impressive growth inhibition. And once again, this is using 1/3 of the dose intensity of mithramycin with concentrations of this RG compound that are potentially achievable in clinical settings. So what I've attempted to demonstrate is that mithramycin is repressing stem cell signaling. It inhibits the malignant phenotype of lung and esophageal cancer cells and mesotheliomas. This growth inhibitory effects appears to be through direct transcriptional inhibition through SP1 depletion, reprogramming by p53 activation, as well as effects on chromatin remodeling. I think that p53 activators are a class of drugs that really could be looked at in the context of malignant pleural mesothelioma and other p53 wild-type tumors. I think precision medicine and pharmacokinetic simulations could enhance the efficacy of epigenomic treatment regimens, and it's a very long and bumpy road from bench to bedside. Thank you.